So our next speaker is Dr. Stephen Robinson, and he's the group leader of the Microbiota and Vascular Health Lab at the Quadram Institute. So Dr. Robinson's research focuses on how the gut microbiome can regulate vascular development and homeostasis.
Thanks for inviting me here today. So having said that my group studies microbiota and vascular biology, I'm not going to talk about vascular biology today. I'm going to talk about the other things that we do. And we're also very interested in how the gut microbiota might regulate diseases such as cancer. So I'm going to take a slightly different approach to the presentation than what Caroline did. I'm going to delve into some of the research that we've actually been doing recently in this arena. So what I'm going to try to do today is answer the question of whether the gut microbiota is a friend or a foe when it comes to breast cancer, developing breast cancer, treating breast cancer. Hopefully you'll come away with the idea that it's a little bit of both, actually. Depending on what aspect you're talking about. And I'm going to tell you the tale in sort of 3 chapters. The first chapter is going to deal with maybe the faux bit of things. The second chapter is going to talk about the friend aspect of things. And then the third chapter is going to basically a single slide about thinking about whether we can use products from the microbiota rather than actual microbiota itself to treat diseases such as cancer. So as sort of a way of introducing the topic, or at least my interest in the topic, I'm going to show you this slide. And it's really just a reminder that our bodies are inhabited by all sorts of different microbes. I'm going to be talking about bacteria, but as Caroline just said, it's not just bacteria. It's fungi, it's viruses, it's archaea, it's a whole host of things. And the reason I really put this slide up is because until a few years ago, I sort of saw myself as basically a conduit for the microbiota that inhabit the surfaces of my body. And I was their transport system. And then, so basically a scaffold to move them from one location to another and to provide them with the nutrients that they need to survive. After all, we are about equal parts bacterial cells and human cells. And then I met this wonderful scientist who you're going to hear from in a little bit later today, who really convinced me that the microbiota is that there's a close association between us as the host and the members of the microbiota that inhabit us, and that the microbiota could really program us to respond and provide benefits to us as a host. And that this might be really important to thinking about how diseases might develop. And more importantly for us, I guess, can you harness that interaction to treat diseases like cancer? So I guess to set the scene in sort of cartoon schematic sort of way, the idea that we have here is that a diverse microbiota is critical for maintaining our host health. And that relates to, Caroline was talking about things like allergies, rheumatoid arthritis, but it also applies to things like developing cancer. And things that disrupt that normal diversity of the microbiota or its normal composition, such as things like antibiotics, which have a huge effect on particularly the bacterial components of the microbiota, disrupting the microbiota with those reagents might cause diseases such as, in this case, breast cancer, or certainly contribute to how it progresses within an individual. So that I'm going to talk about our data in this arena in chapter 1. But the second chapter is really, can we use members of the microbiota to actually treat diseases such as cancer? Can we rebalance the function of the microbiota to restore health and or treat disease? And I've got a little schematic diagram here of a typical probiotic, but I want you to think beyond probiotic, because really what we're talking about here is LBPs, which stands for live biotherapeutic products. So it's a little bit different. It's really using microbiota, using bacteria as drugs to treat disease. So I'll show you some of our data in this arena too in the second chapter. And then in the final chapter, as I mentioned, we'll talk about using kind of what Caroline was talking about. So using, rather than using the bacteria themselves, using their products or their byproducts as potential drugs to treat breast cancer. So I think this slide is really important because it's important to get across to people why we are particularly interested in breast cancer. And I guess this slide is really good at illustrating that the stats with respect to breast cancer, they're not great. So metastatic breast cancer is the most common cause of cancer related to death for women worldwide. So of all of the cancers that kill women worldwide, breast cancer is the number one. Only 15% of women with metastatic breast cancer. So metastatic breast cancer means breast cancer that's spread beyond the breast to other parts of the body. So it's moved from the breast to vital organs like the liver or the bone or the brain. And it's the metastatic disease that kills most people with cancer, particularly breast cancer. So most women with metastatic breast cancer will only survive for less than 5 years. So it doesn't come with a very good overall prognosis. And prolonging the lives of women with breast cancer, metastatic breast cancer, is very expensive. It costs a lot, but more importantly, it's really bad for the quality of life that these people, that these women experience. So it's really important to come up with treatments that are inexpensive so our healthcare systems can deal with them and treat the people that need to be treated. But more importantly, improve the quality of life for these people. So microbiota members, especially good members of the microbiota, offer the, at least potentially, offer the ability to improve the lives of patients in a cheap way. So producing them is going to, we believe, be a lot more economical than some of the very expensive drugs that are on the market. From a personal point of view, we're quite interested in breast cancer because I know a lot of women who have suffered from breast cancer. It sounds a bit, I suppose, cheesy, but it's absolutely true. And unlike a lot of the cancers that are being studied with relationship to the microbiota, the breast is quite distant from the gut. So we're talking about a long distance communication between the bacteria in our guts and the breast cancer that is quite distant from the gut. So a lot of the work that's being done in this arena with respect to cancer is done on things like colorectal cancer. And it's not, I suppose, as difficult to imagine that the bacteria in your gut are going to be affecting the cancer that is developing in your gut because they're physically close to one another. But we're talking about quite, especially in humans, quite a distance between the gut and the actual breast. And then importantly for us, we have really good mouse models of the disease that allow us to delve down into, sort of the mechanisms of what's actually going on. So really good what we call preclinical models of the disease. And we also have a good access because of our close associations with local hospitals to patients that have breast cancer. So we can hopefully in the not too distant future try to translate the findings that we're coming up with in mice, translate those to actual human patients. That's obviously the ultimate goal. So you're probably asking yourself, why do the studies in mice? Why not just go straight to humans? After all, we're trying to treat breast cancer in humans, not breast cancer in mice. Well, as I mentioned a couple of slides ago, we're not trying to develop probiotics that can be used to treat cancer. There is potentially, I'm not really going to touch on it, there is a potential for probiotics to be used to prevent breast cancer, but that's kind of a different aspect of what I'm talking about today. So what we're trying to do is develop, as I mentioned, bugs as drugs, essentially. And that means because we're developing them as drugs, they need to go through the same regulatory pipelines, drug pipelines, that any drug would have to go through. So they have to go through very expensive clinical trials. You need to test them for safety, um, comorbidities, etc. So you can't go directly into a human as easily as you might think. You've got to do all the safety things that you would have to do for any drug. As I mentioned, we're quite interested in the underlying mechanisms of how the microbiota might be regulating the progression, development of breast cancer. And we can't really do those mechanistic studies in humans. It's just not possible. Importantly, the gut microbiota can be easily manipulated in mice, so we can test hypotheses that we might be generating quite readily in a mouse model. And also, I think this is really important when you're thinking about, taking your findings from a mouse and trying to translate them to humans. Sometimes when we're talking about metastatic disease, for example, it might take years for you to see what the outcome of a treatment is in a humans. So that makes it, since science needs to be done reiteratively, that makes it very difficult to do things in humans. So the pathway from inception to disease is a lot faster in mice. So you can do things much more quickly. I hope that makes sense. So chapter 1, it's really, for us, it was the launch of our interest in whether the microbiota might be regulating breast cancer. And we really wanted to establish whether there was a link between the microbiota and breast cancer. So we thought maybe the best way to do this would be to disrupt the microbiota and see what happens in our preclinical mouse models to breast cancer progression in those animals. But we didn't want to just manipulate the microbiota in any way. We wanted to manipulate it in a way that was relevant to patients. So antibiotics immediately pop to mind. They're commonly prescribed to breast cancer patients who are undergoing breast cancer surgery, sort of as a prophylactic to developing infections. They are routinely used in neutropenic patients to prevent catastrophic infections. There is, however, some epidemiological evidence in the literature to suggest that antibiotic exposure increases the incidence of breast cancer. And I guess importantly, from a basic science point of view, these antibiotics can disrupt the microbiome, the microbiota, for quite an extensive period of time. So treat someone with an antibiotic, it might be, in certain individuals, it's up to a year before their microbiota is restored to the levels, the composition, the diversity that it was before that person took the antibiotic. So very briefly, breast cancer in humans is a very complicated disease. There's all sorts of different subtypes of the disease. You've probably heard of triple negative breast cancer. It's a very aggressive breast cancer. For example, it's difficult to treat. You've probably heard of hormone receptor positive breast cancers. They're a little bit easier to treat. They're less aggressive. Basically, what I'm trying to say is that maybe one size does not fit all, but also we can model all of these different types of or subtypes of breast cancer in our mouse models. So basically what we do is we take breast cancer cells that we grow in the laboratory in a tissue culture facility, and we inject them into the breast of the mouse, and we let them grow. We study how they grow, and we see if they've spread to other sites across the body. Mostly what I'm going to be talking about today is their primary growth, so their growth within the breast, or sometimes called the mammary gland of the mouse, rather than their spread to other sites. But I will touch on that briefly. So you're going to see these names pop up in the data slides that I'm going to show you. So these two, the BO1 and the BRPK cells, these represent the most common types of breast cancer that you see. So most women that get breast cancer will have one of these subtypes of cancer. And then I'm going to show you a little bit of data with this additional model as well. And I guess importantly, the profile of the development of the disease is quite different between these different subtypes. And that might be important when you're thinking about trying to translate your findings to a human situation. So this is just a schematic of how we set up the experiments. So in this case, we're taking mice and we're treating them with to begin with, a really harsh cocktail of antibiotics. You can see this is 5 different antibiotics here. And essentially what we were trying to do is wipe out the microbiota in these animals. So almost create a germ-free environment. I think some of our speakers probably later in the day are going to talk about germ-free mice that don't have any microbiota. So we treat these animals with antibiotics for about 5 days. It almost completely wipes out their microbiota, and I'll show you that in a few minutes. And then we inject those breast cancer subtypes into the breast of the animal, and we study how it progresses over time. How does it grow over time? And we continue to treat the animals with the antibiotics over the course of the experiment. So we don't want to establish a regrowth of some of the microbiota members that haven't necessarily been completely killed off by the antibiotics. So when we do that, so I'm showing you how the tumors grow over time. So this is the endpoint growth of these tumors in the mice with those 3 different subtypes of breast cancer that I showed you a few minutes ago. So the volume of the tumors at the end of the experiment is bigger in all of the animals, regardless of the subtype, in the animals that were treated with the antibiotics. So in this case, antibiotic exposure, wiping down the microbiota, has accelerated the growth of the disease in the animal models. Now, I'm not going to show it in any detail, but we've also accelerated the metastasis of the disease, so the spread of the disease to other sites, such as the lung and the bone. And that's really important when you're thinking about what happens in people. So we're making the primary tumor that's growing in the breast grow faster, and it's spreading more quickly to other sites of the body. So I guess short answer here is that antibiotics are not necessarily good. Importantly, though, that is a really harsh cocktail of antibiotics. It wipes out the microbiota, as I mentioned, almost completely. What happens if you use an antibiotic that is used routinely in breast cancer patients? So we canvassed clinicians both within the UK and some of our clinical colleagues in the US, and decided that one of the antibiotics that is used very frequently in breast cancer patients, to use this cefalexin. And in fact, I think I'm not really going to discuss it, but it's really important to think about antibiotic use across different regions of the globe. When we canvassed our clinical colleagues in the US, they were basically, we hand out antibiotics to our patients as if it were like candy. You know, we just do it. We don't even really think about it. And in the context of the data that I'm showing you, that's kind of a scary prospect. But what this slide is showing is that even when you use this antibiotic that doesn't wipe out the microbiota, kills off certain members, bacterial members of the microbiota, we accelerate the growth of the tumor. So it's still bad. Now, as Caroline mentioned, the interface between what's going on in the gut, the microbiota in the gut, and sites distant from the gut is probably an interface with the immune system, right? So, as Caroline mentioned, most of your immune system transits through the gut at some point during its lifetime. It's sampling the bacterial components within the gut, and it's picking up signals from that gut. And it's using that information to not only fight off potential pathogens, but it's using it to pick up signals to fight disease. And in this case, we're talking about breast cancer. Now, I'm not showing you this to go through the complexities of the immune system. I think the last speaker highlighted that very nicely. It's very complex. But basically, what I'm showing the slide is that there's multiple components of the immune system. Dendritic cells that Caroline mentioned, some of the T cells and B cells. All of these cells can play a role in fighting cancer throughout the body. So, for example, I'm showing the dendritic cells here and some of the cytotoxic T cells. The cytotoxic T cells can pick up signals within the gut from the dendritic cells and use that information at distant sites to kill cancer. We spent a lot of time looking at these individual immune cell components in the cancer that was developing in the mice from animals that were and were not receiving antibiotics. And we did not see any changes in any of the classical components of the immune system that you might think would be associated with the responses that we were seeing. However, through a very complicated set of experiments that I won't go into, it took us months and months and months to find an immune cell population that was actually being altered by the use of the antibiotics. And we came across this sort of obscure immune population of immune cells called mast cells. You've probably heard of them. They're often associated with fighting off parasites in the gut, like worms, for example. They're associated with allergic reactions to things in your skin. So if you, for example, get pricked with something that you might be a little bit allergic to, you'll get an immediate immune response. You'll get a welt, you'll get a red swelling, and that response is mediated by the factors that these immune cells, these mast cells, release into the local environment. So it turned out, and I guess the reason that we persisted in looking for immune cell populations is because we're stubborn. Scientists can be stubborn. And I refuse to believe that there wasn't some sort of immune interface between changes in the microbiota and the breast cancer. So we came across these mast cells. They do play a really important role. When we counted the mast cells in the tumors from mice that were treated with antibiotics, they were significantly elevated. So you can see these purple dots here are the mast cells. And there's many, many more of them in the tumor from the animal treated with antibiotics compared to those animals that weren't treated with antibiotics. And this was true across the spectrum of the disease. It didn't really matter what antibiotic we were using. And it didn't really matter what type of disease we were looking at. Every time we treated with antibiotics, we saw an elevation of the immune cells, the mast cells in the tumors from the antibiotic treated animals. So that is just a correlation, right? You treat with antibiotics, you see an elevated level of mast cells in the tumors from these animals. You'd really like to know that those mast cells are playing an important role. So we did an experiment where we prevented the mast cells from releasing those little chemicals into the surroundings to see what would happen. If we set up the experiment in exactly the same way as I showed you before, treat with antibiotics, put in the tumor cells, let the tumors grow over time, and then as they reach a fairly significant size, we treat it with this drug called cromolyn. And what cromolyn does is it prevents mast cells from releasing their contents. So they don't release those little factors that lead to the welt in your skin. When we do that, we don't get the increased growth of the tumors in the animals that were previously treated with antibiotics. So that tells us that the mast cells are playing a really important role in mediating the effects of the antibiotics, right? So there's some sort of interface between the microbiota in the gut and the function of the mast cells within the tumor. We still don't know what that interface is, but we do know that the microbiota is somehow communicating with those mast cells. And if you're thinking about developing a drug, for example, it gives you a potential target for developing a drug. So it might be that in a human that has seen antibiotic exposure and their mast cells have gone haywire, you could prevent those mast cells from releasing their contents and having this negative effect. Now, that's a huge extrapolation that needs to be tested on multiple levels, but it gives you a hypothesis and something to test. So I want to turn, so that kind of suggests that if you, the faux part of the story is the microbiota can play a really important role in limiting progression of diseases such as breast cancer, and that if you treat it with antibiotic or you disturb it with antibiotics, you can have a deleterious effect on the progression of the disease. But that can happen in two ways. It could be that what you're doing with the antibiotics is that you're allowing amplification of what we call a pathobiont. So something that isn't necessarily a pathogen, but a bacterial member of the microbiota that, if it's present in the wrong amount or in the wrong ratio compared to the other members of the microbiota, it can play a detrimental role. So that's a formal possibility, that the antibiotics were killing off most of the microbiota, but not some of these pathobionts. The other potential thing that could be happening is the antibiotics are eliminating bacteria in the gut that play a protective role, that are responsible for limiting the progression of the disease. And this experiment shows you that we think that what the antibiotics are doing is they're getting rid of a friend, someone, a bacterial component, or a number of bacteria in the gut that are playing a protective role. And to make a complicated experiment simple, I guess what we're showing here is if you take the bacterial contents of a mouse that has been treated with antibiotics and you transfer it to an animal that has not received antibiotics, you don't accelerate the progression of the disease, right? If you do it the other way, though, you do. So essentially, if you take poo, so mice are very good at eating their own poo. So if you put them in a cage that has poo on the bottom of the cage, they'll eat the poo. It's called coprophagy. A lot of rodents do the same sort of thing. And basically what this is showing you is if mice that have been treated with antibiotics eat the poo of control animals, it reduces the growth of the tumors in those animals. So it looks like you're adding back a friend, right? So what that means, though, and I think this is the important message from those experiments, is that if you know who that member is, who that bacterial member is, you can add it back to a mouse or a human that's been treated with antibiotics and prevent the negative effects of the antibiotic. So that kind of switches us into chapter 2, and that is, can we develop live biotherapeutic products, or can we add back bacterial members that restore the normal microbiota, or bacterial species that might train the immune system to function better in its attempts to kill off cancer cells or limit the progression of, in this case, breast cancer. So our attempts at developing LBPs, or live biotherapeutic products, is chapter 2. So through a very complicated set of experiments, you can actually examine the different types of bacteria that are present in the guts of these animals that have been treated with antibiotics. So basically, you isolate all of the bacterial components of the gut microbiota from these animals, and you can sequence the DNA that's there, and you can determine all of the different species of bacteria are there. So this is just kind of pie charts representing some of the major classes of bacteria that you see in the gut. And you can see from the animals that were treated with a very harsh cocktail of antibiotics, you pretty much wipe out all of the bacterial components. You're left with mostly viruses that antibiotics don't touch. It's a lot less of a harsh effect in the animals that have been treated with a clinically relevant antibiotic. So basically what we wanted to do is to look whether there was some sort of commonality between the animals that were treated with a harsh cocktail of antibiotics and those that were treated with the clinically relevant antibiotic. And we came up with a number of species of bacteria that looked like they were being affected. So their presence in the gut microbiota was being reduced by the antibiotics. And we hypothesized that it's, getting rid of one or more of these that might be responsible for, because of the action of the antibiotics, responsible for the progression of the breast cancer in these animals. And we became really interested in this one called Faecalibaculum rodentium, because at about the same time we were doing these studies, other people were showing that, I'm going to call it F. rodentium for ease of pronunciation, could play a protective role in gut cancer, so colorectal cancer. So down here is just the human equivalent of this bacteria is called Holdemanella biformis, so H. biformis. So we took a closer look at the presence of F. rodentium in the gut microbiota of our animal models, and it is significantly reduced in animals treated with a harsh cocktail of antibiotics and those treated with the clinically relevant antibiotic. And I guess importantly for us, when we add F. rodentium back to our animal models, we reduce the growth of the breast cancer in these animals. So that suggests that F. rodentium is playing a very important role in limiting the progression of the disease. So we're adding back a friend. And that opens up, I guess the caveat here, though, is that we're just returning the growth of the breast cancer back to that we would see in control animals. So we're not, suppressing the progression of the disease, but we're preventing it from being accelerated by the effects of the antibiotic. And that's important. But most importantly, it opens up the possibility that we could put beneficial bacteria back into the gut of breast cancer patients and alter the course of their disease. So the questions that we're left with, I suppose, are what is F. rodentium doing? Is it producing metabolites that are programming the immune system? Do we see the same effects with the human homologue, the H. biformis? And can we probe the microbiota of human patients with breast cancer to see if we see any similarities between our mouse models and what's going on in people? So, unfortunately, just before the pandemic hit, we started a human cohort study where we are trying to collect fecal samples from breast cancer patients. It's called the BEAM study. It's being run out of the Norfolk and Norwich University Hospital, and hopefully very soon out of the James Paget Hospital in Great Yarmouth. And basically what we're trying to do is determine what happens to the microbiota of humans with breast cancer over the course of their treatment. Do things like the antibiotics that they'll be receiving or the chemotherapies that they'll be receiving change their microbiota? And can we make any correlations with what we see in animal models, in our animal models? I said, unfortunately, it started just before the pandemic. Unfortunately, because the pandemic's kind of reduced a number of patients that we can recruit onto the cohort study. But as things start to return to normal, we're beginning to hopefully recruit more breast cancer patients onto the study. So that, I guess, is a really good segue into showing you the studies where we are using what are sometimes considered probiotic members of the microbiota to actually treat breast cancer. So not just reduce things in patients that have been exposed to antibiotics, but use the bacteria to reduce the progression of the disease. So putting the specific species of bacteria into the gut to program the immune system to work better. So this is just showing you some of the early studies that we did in this arena. The setup is a little bit different. We put the breast cancer tumor cells into the animals. We let them grow to a certain size, and then we start to treat with our LBP, our live bacteria. And in this case, we were putting in a cocktail of four different bacterial species and seeing what it did to the growth of the primary tumors in the breast. So here you're looking at the volume of the tumors at the end of the experiment. And in both of these hormone positive subtypes, we see the primary tumors the growth of the primary tumors is significantly reduced. And importantly, so one of the weird things about breast cancer is that it can metastasize or spread to distant sites very early on in the course of the disease. And then those breast cancer cells sit at those distant sites, waiting for the opportunity to grow later in life. Places like the gut, the lung, the liver, the brain, the bone marrow, those early disseminated or spreading cells can sit there for a really long time. So importantly, our live bacteria are also reducing the spread of the cancer cells to the lung in these animal models. And this is just showing you we can analyze, look for the cancer cells in the lung, and we see a lot fewer in the animals that have been treated with our live bacteria. So we have taken that cocktail of 4 different bacteria, and we've tested each of the individual bacterial species in isolation. 3 of those on their own. So a single bacteria do very similar things to the cocktail. And we've taken one of those. It's kind of our favorite bacterial species for reasons I won't really go into. And we've tested it in a lot of detail to see what it's actually doing to the immune system in our animal models. So what is it doing to the cancer-fighting immune system in our animal models? And it's actually, I'm going to skip a couple of slides to show you what it's doing to the immune system because I'm starting to run out of time. So the idea is here that we really want to program the cancer-fighting immune system to work better, right? And we can do that through, we hope, by putting in good bacteria and telling the cells of the immune system to kill off the cancer cells a little bit better. And that's exactly what we're seeing. So when we look at the immune system in the tumors from our animals that have been treated with bacteria, we're seeing a good response of immune cells that would kill off the cancer cells. And this is showing you that those immune cells are actually producing enzymes that are responsible for perforating and killing off the cancer cells. So that's a good thing. We're a step further in developing bugs as drugs. And then finally, in the final third chapter slide, that's live bacteria. And as Caroline was talking about, you don't always necessarily have the opportunity to use live bacteria. You might want to use their products. So this isn't breast cancer anymore. It's actually melanoma, but we're developing products from these bacteria that can be injected directly into the animals, and it can change the course of the growth of these melanoma cells. So not the bacteria themselves, but products from the bacteria. And that gets around potentially the concept of these bacteria needing to colonize the gut to be there live, very similar to some of the things that we were talking about in the last talk. And then I guess that just leads me to summarize, to say that the gut breast cancer axis is important. It can regulate the function of mast cells, so we've got a potential target there. And we know commensal microbiota members hold promise for improving treatments of some types of breast cancers. So really what we're trying to do is take the bacteria we've developing and using in our mouse models and try to translate those to people with breast cancer. And then like Caroline, that just leads me to thank all of the people that have done this work. These are the people in my lab who have done the work, and then the people who actually, the organizations that actually fund us to do the work. So I'm happy to take any questions, although we're almost out of time.
Thank you, Stephen. I'm sure we can squeeze in a couple of questions. That was a really interesting talk about such an important subject. We have had some questions on both Discord and on the YouTube channel. So let's start with one from Fred. And he asks, why are breast cancer patients given antibiotics in the first place?
Well, I think as I tried to say, I mean, a lot of times we're not saying don't give breast cancer patients antibiotics. Frequently, because of the chemotherapies that breast cancer patients are undergoing, their immune system's not functioning very well anyway to fight off infections. So those antibiotics are really essential for preserving life, preventing sepsis, for example. So, giving those antibiotics can be essential for, the breast cancer patient not dying from sepsis. But if we know what the antibiotics are doing, we can try to counteract those effects.
Great. On YouTube, I think Joe asks, do you have any thoughts on the effects of the microbiota on bile's effect on breast cancer? Is that something you might have a comment on?
Say that again. On
the effect of the microbiota on bile's effect on breast cancer. Any thoughts on bile?
On bile, like so bile acids and stuff.
Yeah.
Yes. So, one of the things that we're looking at. So one of the things that we would really like to know, for example, is what metabolites are changing when we treat our animal models with antibiotics. And one of the things that changes dramatically with antibiotics is the bile acids that you see emanating from the gut. And they're quite elevated with respect to the antibiotic treatment. So, one of the hypotheses is that those bile acids may be playing a detrimental role as well. So that's something that we're actually testing at the moment.
Oh, fascinating. Very quickly, just to finish up, Amy and Irene are both asking similar questions about, do you think that in the future, more patients will be prescribed probiotics along with antibiotics? And
Yes.
what's holding it back?
Yes, in a nutshell, I think is the answer. If nothing else, even if we don't get to a point where the probiotics are actually affecting the development of the cancer itself, one of the nice things that Caroline mentioned that probiotics do is that they can restore the integrity of the gut, right? And a lot of the chemotherapeutic drugs that cancer patients are given really disrupt that integrity of the gut. And they get things like diarrhea and or constipation because of the disruption of the integrity. So if nothing else, the probiotics are really good at making cancer patients feel better because they don't get those sort of gastrointestinal side effects.
And that's a really positive note to finish the quick discussion on. Stephen, would it be possible for you to pop onto Discord if anyone has any questions to follow up there?
Yep, no problem.
And I'm sure people would love to see a copy of your slides or any follow-up links that you could provide.
Yep, no problem.